Well, good afternoon, everybody, and thank you to the Hamilton Committee for giving me this opportunity to tell you about my doctoral research. But before we dive into that, let's reel off a little bit cancer, which, as you may know already, is an evolutionary process. So in a very simplified way, cancer starts with one or a bunch of cells mutating and starting to divide with no control, making an heterogeneous primary tumor. Eventually, one cell of that primary tumor will acquire the ability, well, let's say the mutations, to leave the primary tumor and spread to other parts of the body where it will make new tumors. This process, we know it as metastasis, and it is the leading cause of cancer deaths nowadays in our population. Another key concept of cancer for this talk is that the cancer is restricted to the life of the host. In other words, if the patient dies, the cancer will die with it. However, this is not always the case, as there are some cancers that have the ability to live not only to spread to other parts of the body, but to leave the organism where it was originated and spread to other individuals. These are known as clonal transmissible cancers or contagious cancers, and there are three well-known cases in nature. One of them is affecting dogs, another one is affecting Tasmanian devils, and the third one is particularly frequent under the sea, more specifically affecting these animals, which are bivalves and live buried under the sun. So the bivalves had leukemias, a leukemia-like cancer, that were started to be reported in the 60s or 70s of last year, but it was not till 2015 when the contagious nature of these leukemia-like cancers was reported. And that was done by genetic studies. So phylogenetic analysis of these cancers is crucial to untangle if a cancer is or not contagious. How? Well, if you take several individuals and we sequence the cancer cells and the match normal or healthy cells of those individuals, and then we build the phylogeny with the genomic data, we will find that the cancer cells will cluster close to the healthy cells of the individual where they were originated, as they share the common origin. However, if we are looking at contagious cancer, the outcome might be very different, as all the cancer cells that are present in different individuals come from the same or former individual or original individual. Therefore, they will cluster together in the phylogeny. And with all of this in mind, let's now see the story of the evolution of two transmissible leukemias colonizing the coasts of Europe. So cockles, Therastoderm medullae, are distributed in the Atlantic coasts of Europe from northwest Africa to eastern Russia. And from all the cockles that we collected, we detected cancer in just 5.7%. So that is the prevalence. And if you compare it with human cancers, this is a very high figure. However, all those cancer samples were detected in Southern European countries, despite the effort made in sampling Northern countries. In terms of phenotype, our study supports what was already reported in literature. So there are two different phenotypes based on cell size and morphology that are called type A and type B. Also, in 2016, it was published a study in Nature about the contagious cancers of cockles, and it revealed that two independent contagious cancers in cockles were spreading among the populations. So we repeated that with our extensive sampling with more locations, and we corroborated the same story. We also had a look at the satellite markers because they are very good candidates, as they are long-term conserved, and they are also have rapid copy number changes. And what we found bioinformatically and then validated through fish proofs in the lab one of the remarkable findings was that these contagious cancer lineages have to be very ancient, as there were several satellites that expanded after the contagious cancer lineages appeared, and therefore are present in nowadays Cockle's healthy populations. If you're having a look at these chromosomal images, you may already be thinking on my next slide. Cockles have a common diploid chromosome number of 38. However, when we have a look at the cancer cells, we have a high abundance of small chromosomes and a very unusual ploidy. Having this in mind, it led us to analyzing the copy number profiles of these cancers using the whole genome sequencing data. And what you can see here is a global pattern of copy number alterations along all the chromosomes, with a 10 of a tetraploid state. And that happened for the contagious cancer lineage 1 and also for the second one. So this is suggesting that for these cancer lineages, it was a very important early event, a whole genome duplication as a driver of the evolution of these cancers. Combining this copy number data with a structural variation and also selection analysis, we went into the hunt for driver candidates to understand the early evolution of these contagious cancers. And these 4 genes that you're seeing in the slide right now stand out from our analysis. So as you can see, we found a deletion in the tumor suppressor gene, and we found amplifications in the oncogenes as expected. And all these genes have already been reported as drivers in tumors in multiple cancers. So they were very good candidates. We continued further, and we also had a look at the RNA data, so the transcriptomic data, to look at the expression that actually validated what we were looking at the genomic level. And even more, in the case of the MDM2 gene, we also saw overexpression of that gene in the contagious cancer lineage 2 that we couldn't find in the genomic data, suggesting that this gene might have played a very important role as a driver in the early evolution of these contagious cancers. And the last bit of this presentation is for the mitogenome, so that piece of DNA that we have in the powerhouses of the cells. So we built a phylogeny, we deconvoluted the genomes and we built a phylogeny, and as expected, because we already knew that it was contagious, tumor genomes mitogenomes were clustering together. However, instead of looking at 2 contagious cancer lineages, we ended up with 9 cancer lineages, as you can see highlighted in green. So our first question was like, are these cancer lineages independent? So we ran some topology testing that is basically constraining a phylogenetic tree and looking for the probability, and all the different methods used confirmed that these cancer lineages are independent. So the next step was to combine this data with the phenotypic and the nuclear profiles that we had. And as you can see, all the cancer lineages belong to whether type A or type B. There is not a single lineage that it has a mixture of different cancer types. So with all that in mind, we started to understand this story, what is happening here. So we hypothesized that as in cancer, there is a high rate of high mutation rate. In order to avoid the decline in fitness, because as you know, the mitochondrial DNA is very important for the cells, the cancer cells of these contagious cancer lineages that are very ancient find a way to overcome this issue. And that was to capture mitochondria from healthy cells and therefore overcome the decline of their mitochondrial DNA. So as you may imagine, there are many other things that I could tell you about the evolution of these contagious cancers, but today I have shown you some of the facts or evidences that we have that support that these contagious cancers that are very ancient have a high tolerance to extreme genomic instability. And if you're curious and you want to read a little bit more about these cancers, I invite you to read my thesis or this preprint that is already available and is going to be soon published in Nature Cancer, that tells all the things that I have shared with you today and some others that I didn't have the time to do. And last but not least, I would like to thank the organizing committee of this conference, the lab where I was working, the funders and the institutions for the support to do this work, and of course, all the cockles that gave their life so we could do this science. So thank you very much.
Great, well, thank you. And we again have time for multiple questions. So get your hands up and we'll answer them in the order we get them. All right, so Walid, you are first this time as well.
Thanks, Mike. Alicia, very, very cool system, very cool results. Simple question on, I was surprised with what you were talking about at the end about the possible horizontal transfer. Have you even observed any kind of that kind of mechanism going on in this system? And two, have you ever thought of any possibility of nuclear and mitochondrial genome conflict within the cell that might be also driving some processes related to that?
Hi, thank you for your words and also for the question. Mitochondrial transfer has already been reported in human cancers and other types of cancers, and it has also been validated in vitro and in vivo. So it was not a surprising or a novel process that we have found here. It's just that we have found it in this contagious cancer that we didn't know that it existed before. I agree with you. There might be some conflict with the nuclear DNA, as the nuclear DNA it appears to be very ancient, and the mitochondrial genome might be captured nowadays, so there might be some conflicts. However, this is still an ongoing project, and we try to continue analyzing the genomic data for the nuclear SNVs, and as soon as we find some data supporting some conflict or not conflicts, we will publish straightforward. But thank you for the noticing.
All right, and Isra, you can ask a question as well. Ryan, if you'd like to start sharing your screen, just get things set up, that's fine too.
That was a wonderful talk, Alicia. Really cool system. I wanted to ask you about the unusual ploidy or sort of broken genomes that you see. Is that something that declines over the lifetime of the cockle or over multiple generations? Do you see the number of those broken chromosomes? Does that change?
Well, isn't it amazing, the ploidies that we are observing? I mean, I was shocked when I saw that this the first time. So the thing that is weird here is that the other transmissible cancers that were reported in dogs or Tasmanian devils, they don't show this. So this is something that is particular from bivalves, and we have seen it in cockles in particular. And we don't know how is the cancer cell able to survive with that amount of chromosomes? Sometimes they are up to 300 chromosomes in a single nuclei.
Sorry, do you see any exchange of those chromosomes? So you've shown that there is a horizontal transfer of the mitochondrial DNA. Is it possible that they are sort of selfish genetic elements that are traveling?
So we are doing an analysis of line elements, so to see how mobile elements are working in these chromosomes, because we do think they play an important role. However, what you are saying, there is a very interesting hypothesis of capturing the also chromosomes from the host cells. Could be happening. We haven't looked at it right now, at least not yet, because as you may know, when we collect the cancer cell, it is in a host, but maybe the capture from mitochondrial or nuclear DNA was in the previous host, and we don't have that previous host.
So I'm going to have to cut you off here so that Ryan gets to start in about 10 seconds.